What's up, Bio Penguins? So it was asked for me to tell you about the things that my students did wrong on their mock exam, as well as I'm going to share some tips and tricks for the AP Bio FRQs. So first thing is all about experimental design. So being able to know what the difference is between a negative control and a positive control. And so here's a cool graphic that I found that might help out. So the big difference is that your positive control, you know what's going to happen with the experiment, as well as it does give you some type of response. And so it's used to test the validity of the experiment, versus the negative control does not get whatever the treatment group is. And so it's going to kind of ensure that there's no response to the actual treatment. So it helps you to figure out the influence of those external factors. The next thing is about independent versus dependent variables. So your independent variable is what the scientist is changing and what the researcher is doing. And the dependent variable is what changes because of that change. This is oftentimes just given in the prompt. So when you're reading through the prompt and it says the researcher was testing the effects of blah, blah, blah on blah, 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 you know that the dependent variable is what they were, of course, the data they were getting, while the independent variable was what they actually were changing. Speaking of controls, being able to determine kind of what the purpose of the control is. So a lot of times students are saying that the control is used to compare your results. We'll be very specific about your experiment, about what specifically the control is doing. It is allowing you to see the effects of XYZ chemical. It is allowing you to see the effects of XYZ independent variable. Null hypothesis was kind of hard for students. So whenever you're doing null hypothesis, it's always going to be that independent variable has no effect on dependent variable. Okay, again, you want to be very specific about your experiment. Graphing was hard for students. So your graph can usually be 3 to 4 points, 3 points for the graph, 1 point for analysis of that graph. So completing a graph is oftentimes going to get you some of those easier points. So one, figuring out what kind of graph it is. It's a line graph if we have some type of linear data. It's a bar graph if we're looking at categorical data. It's a scatter plot if you have usually 3 different things you're looking at. So there's like different individuals, and then you're comparing how this has an effect on that. And so that's usually going to be our scatter plots. No matter which graph you choose to do, you want to make sure that you include your error bars. So whenever you're looking at the data in the table, it's going to have the mean being the first number, and then there's going to be a plus or minus and then another number. That plus or minus is telling you your error bar. You're going to graph above and below that by just drawing a line to those points. Scaling your axis, so you want to count by a number that's logical, something that you can easily count by, and you do not have to label every single box. So like, for example, if each box is equal to 2, then every 5 boxes you can just label 10. Another issue my students had was defining their terms. So they assumed that the readers knew what was happening. And so we as readers can't assume anything. So if you are saying that something has more fitness, what does that mean? So if you're trying to say that it's more fit in your explanation, then you would explain that, oh, there's going to be more individuals that will survive, and we'll see more individuals that are going to be able to pass on that trait to their next generation. So you need to make sure that you are very distinct in explaining those things. Reading your questions very carefully was another thing. So students were giving overall trends to graphs instead of explaining what the question asked. So the question was asking like for a certain segment of time. So make sure that you are answering the questions that are being asked of you. So let's talk overall tips and tricks. So you have 90 minutes to do 6 FRQs. There are 2 long ones and 4 short ones. So for me, I tell you to go ahead and take the first 5 minutes of the exam and look through all of the questions and go ahead and kind of renumber them, pick it out. So you have 1 long and then 2 shorts and then another long and then another 2 shorts. But renumber them and do the things you know in the order that you know them. You want to spend about 20 minutes on each of the long FRQs and about 8 minutes on each of the short FRQs. So after you do that, you should have another about 10 minutes left that you can go back to any questions that you might not have finished, or you're wanting to sit there and kind of think about more things you could add, or if you want to reread your responses, just make sure that things are logical. Sometimes as you're writing, you skip information that you're meaning to include. Another thing is that I've noticed this every year is that students freak out because they flip the page and they've just been working on question 3, and at the top of the page it says start response to question 3. So then they're like, oh, I have to erase, basically scratch out everything that I've done and rewrite it. Please do not do this. Just write a note on question 3, see question 2 for question 3, and your readers have access to your entire book and will be able to find your response. So it's helpful if you label the sections like A, B, C, D. You do not have to answer these in order. You can answer them out of order. So if you know C first, answer C, then maybe A, then D, then B. It doesn't honestly matter, but it is helpful for your readers if you go ahead and label the sections. If you make an error and you want to get rid of something, just put a single line through it. If it's an entire paragraph that you're getting rid of, just put a big X through it. You do not have to put a line through every individual little section. The readers are getting a scanned copy of your response, so make sure that the pen that you're using is not bleeding, as well as make sure that you're not using an erasable pen. Erasable pens are erasing by heat, and so the heat in transit from your school to the warehouse, and then maybe sitting at the warehouse for a while, could cause your responses to erase. This video is longer than I expected it to be, so I'm so sorry, y'all, and I'm going to go ahead and just stop. Let me know if you need anything else in the comments. So AP Bio Penguins, just assessed by y'all.